Hello students, let us see how the integration of metabolism happens, and basically let us see the concepts, the key concepts and the involvement of various organs in metabolism. To introduce this lesson to you, integration or interrelationship of metabolism is the coordination of metabolism of fat, protein and carbohydrate in order to maintain adequate levels of ATP and glucose. The purpose of energy metabolism from these three macromolecules is to provide energy when demanded and preserving energy when not needed. As no single organ is metabolically independent, this integration is achieved by different specialized organs, namely liver, adipose tissue, brain and muscle, which contribute to accomplish the task by their own specialized functions. Coming to the objectives, we are going to see in this lesson the generalities and characteristic features of the integrated metabolism, the 7 major metabolic pathways that fuel the body and how they are metabolically interconnected, the major metabolically active organs which could function in a synchronized fashion, and the impact of 3 different states of feeding on metabolism. So let us see the salient features of interrelationship. The currency of and crucial metabolite of interrelationship. It is well known that the currency of energy is ATP and its hydrolysis. The only 2 major sources of ATP are TCA cycle electron transport and of course glycolysis. Mitochondrial electron transport chain fueled by the TCA cycle produce much of ATP molecules, which in turn depend on the supply of oxygen. Cells that lack mitochondria like erythrocytes do not have TCA cycle or oxidative phosphorylation and hence use glucose, the central metabolite, as a sole source of energy. In the absence of constant supply of this crucial metabolite, which again I call it CM, the cell cannot survive. Brain is the one such organ that drastically depends on glucose metabolism for energy. However, it also could use alternative fuels other than this crucial metabolite. The currency ATP and the crucial metabolite need to be stored in the cellular process of energy metabolism. Fat is the major storage source of ATP, the energy currency, and glycogen is the storage form for glucose, the CM. Protein, yet another molecule, serves as useful storage source for both energy and glucose. Coming to the reducing equivalents in metabolism, in an anabolic pathway of any metabolism, the metabolites are further reduced from their precursors. This necessitates a reducing power along with ATP. High potential electrons serve as driving force for such reactions and are usually supplied by nicotinamide adenosine dinucleotide phosphate. Pentose pathway, of course, is the major provider of NADPH. The metabolic pathways which produce energy, namely ATP and reducing equivalent NADPH, also provide building blocks for the synthesis of more complex molecules. Coming to the signals which are primary and secondary and their states in interrelationship. The storage forms respond to different signals for executing various cellular processes during different metabolic states. The 3 major primary signals are insulin, glucagon and epinephrine in metabolism of major metabolic states such as feeding, fasting, starvation, diabetes and excitement. Insulin and glucagon are antagonistic in their action in regulating the blood glucose levels. These signals utilize and liberate energy respectively. Epinephrine is another major signal that is similar to glucagon and functions where there is an intermediate demand for energy. There are also secondary signals which could be grouped as 4 classes. namely low energy, high energy, low glucose and high glucose. ATP, fatty acids, citrate, NADH, acetyl-CoA form high energy signals, while cyclic AMP, ADP, AMP and phosphate form the low energy signals. Fructose 2,6-biphosphate and glucose 6-phosphate form the high glucose signals, while cyclic AMP forms the low glucose signal. Not all of them alter the activities of all enzymes or pathways, and they exert an effect in the direction indicated by the type of the signal. For example, fructose 2,6-biphosphate is a signal for the presence of glucose. Hence, it is anticipated that the increase in the levels of fructose 2,6-biphosphate would result in increased rate of glycolysis and increased fatty acid synthesis. But fructose 2,6-biphosphate can only promote glycolysis and decrease gluconeogenesis by affecting the activities of phosphofructokinase and fructose 1,6-biphosphatase. It cannot directly affect the activities of other enzymes and is a local signal. It should also be understood that the effectors such as ATP, that is a high energy signal, would inhibit phosphofructokinase, a key regulatory enzyme in glycolysis, and thereby inhibit the energy production from glycolytic pathway. However, this futile cycle could lead to rapid changes in flux through cycle, resulting in a more sensitive control. Such futile cycles are minimized by signals which turn on a synthetic pathway and thus turn off the opposite degradative pathway, which is just shown in this figure. Low energy levels stimulate lipolysis and glycolysis, while low glucose levels stimulate protein degradation and thus gluconeogenesis. Activation and inactivation of enzymes, their allosteric interactions and levels are recurring motifs in metabolism. Phosphorylation or dephosphorylation occur in response to cyclic AMP levels. During low energy and low glucose levels, there is an increased activity of CAMP dependent protein kinase and thus enzymes are phosphorylated. During high energy or high glucose levels, dephosphorylation occurs. The fates of molecules are decided by compartments of cells such as cytosol or mitochondria in which they are present. Coming to the interrelationship of major metabolic pathways. The major metabolic pathways, as I mentioned in the objectives, are glycolysis, gluconeogenesis, glycogen metabolism, fatty acid metabolism, citric acid cycle, oxidative phosphorylation, and of course amino acid metabolism. Coming to the first pathway, namely glycolysis. The purpose of this major degradative pathway is not only to provide ATP but also to provide carbon skeleton for biosynthetic pathway whose rate is dependent on ATP-AMP ratio. The key regulatory enzyme phosphofructokinase is subjected to regulation by levels of fructose 2,6-biphosphate, which in turn depends on activities and levels of kinase and phosphatase. As I mentioned earlier, low glucose levels promote glucagon-triggered signaling that cause an activation of phosphatase and inhibition of kinase, leading to low fructose 2,6-biphosphate and thus its target phosphofructokinase. This decline in phosphofructokinase activity causes slowing of glycolysis. The remaining glucose enters blood for utilization by other tissues as shown in this figure. Coming to the second cycle, citric acid cycle and oxidative phosphorylation. This is a classical integrated catabolic pathway for all the molecules. There is a tight coupling of electron transfer and ATP production. The respiratory ratio which ensures the balanced rate of this pathway and demand for ATP. An excess of ATP would diminish the activities of isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. This classical catabolic cycle also has anabolic role along with the activity of pyruvate carboxylase by providing succinyl-CoA for formation of porphyrins and citrate for the synthesis of fatty acids. Coming to the third pathway, that is pentose phosphate pathway. This occurs in cytosol and it consists of two stages. 1. Oxidative decarboxylation of glucose 6-phosphate and 2. Non-oxidative catabolism of pentoses. While former supplies NADPH for reductive biosynthesis and ribose 5-phosphate for synthesis of nucleotides, the non-oxidative part could either reduce riboses into catabolic glycolysis or generate riboses from glycolytic intermediates for anabolic pathway. Gluconeogenesis. This distinctive pathway ensures the synthesis of glucose in liver and kidneys from non-carbohydrate precursors which use two different compartments of cells. Oxaloacetate is generated in mitochondria and then enters cytosol to form phosphoenol pyruvate. Another distinctive feature is the presence of two hydrolytic steps which circumvent the irreversible reactions of glycolysis. Fructose 1,6-biphosphatase, a vital enzyme in gluconeogenesis, is inhibited by AMP and activated by citrate. It could be interesting to know that these molecules have opposing effect on the enzyme in glycolysis. Abundant glucose and thus fructose 2,6-biphosphate would result as a consequence of glucogenic pathway as shown here in this figure. Coming to the fifth cycle, glycogen synthesis and degradation. This is yet another metabolism that follows generalities of integration of metabolism. Glycogen synthesis and degradation are regulated by primary signals, secondary signals, phosphorylation, dephosphorylation, and of course non-covalent allosteric interactions of two enzymes, namely phosphorylase. Phosphorylase is glycogen degrading enzyme which yields, as shown in this figure, the glucose 1,6-phosphate that gets converted to glucose 6-phosphate. UDP glucose is the active intermediate in glycogen synthesis and is formed from glucose 1-phosphate and UTP. Glycogen synthase catalyzes the transfer of UDP glucose to the terminal glucose residue. That is, synthase is inactive when phosphorylase is active and vice versa occurs. Coming to the sixth phase of metabolism, that is the cycle, namely fatty acid synthesis and degradation. This serves as one more classical example of compartment concept of integration of metabolism. From the mitochondria, acetyl groups are carried to cytosol as citrate by citrate malate shuttle and gets converted to acetyl-CoA. Citrate in the cytosol stimulates acetyl-CoA carboxylase producing malonyl-CoA. When ATP and acetyl-CoA are in surplus, the citrate would increase, thus accelerating the fatty acid synthesis. On the other hand, carnitine transports fatty acids into mitochondria for beta oxidation. The resulting acetyl-CoA enters TCA cycle and might also enter ketogenetic pathway. As all of you know, FADH2 and NADH are formed in the beta oxidation and transfer their electrons to what is called as electron transport chain. It is worthwhile to know that the rate of degradation of fatty acid is coupled to the demand of ATP levels and could be inhibited by acetylcarnitine by carnitine acyl transferase. Coming to the concept of tissue cooperation, let us take for example in liver. Liver uptakes glucoses from diet via intestinal absorption and metabolizes into glucose 1-phosphate for meeting the energy demands. leaving some glucose back in the blood. It also governs the production of glucose by promoting glycogenolysis and by gluconeogenesis in response to the energy levels and ATP demand. The precursors for gluconeogenesis are lactate and alanine from muscle, glycerol from adipose tissue, and glucogenic amino acids from the diet. In fed state, fatty acids from diet and fatty acids synthesized in liver are released into circulation as low density lipoprotein. But in the fasting state, fatty acids are converted by liver into ketone bodies. It could be recalled that carnitine acyl transferase is inhibited by malonyl-CoA, and thus if the latter is present in abundance, the transport of fatty acids is abolished from entering mitochondrial beta oxidation and ketogenesis. Rather, they are diverted and get exported into adipose tissue for incorporation into triacylglycerols. If the levels of malonyl-CoA are low, fatty acids are liberated from adipose tissue into mitochondrial matrix. The liver also plays an important role in metabolizing dietary amino acids. Owing to the low km of amino-acyl tRNA synthetases when compared to that of catabolic enzymes, protein synthesis is promoted in preference to the degradation. In degradation, the first catabolic step for amino acids is the removal of nitrogen, which is processed into urea. It is interesting to note that liver could not use branched amino acids and acetoacetate as fuels. Of course, it derives energy from alpha keto acids and shuns the fuels to muscle and to brain. Coming to the second organ or tissue, muscle. Muscle, unlike brain, stores about 3/4 of glycogen in the body, which is converted to glucose 1-phosphate. However, muscle joins brain in lacking glucose 6-phosphatase and thus do not export glucose. The organ prefers to retain glucose for its activity as shown in the table. Core's cycle is shown in the figure, which is the major energy cycle that occurs in an active muscle forming pyruvate, and the rate exceeds that of TCA cycle. Much of the pyruvate is converted into lactate and some amount of lactate gets exported to liver. Further, the active muscle also produces alanine to get rid of nitrogen in the form of alanine. The difference from the muscle, liver could absorb and transaminate branched amino acids, but it cannot produce urea. Liver takes up the released alanine, removes nitrogen as urea, and proceed with gluconeogenesis. Unlike the active skeletal muscles, the major fuel for resting muscles is fatty acid. In the case of heart muscle, there are no glycogen reserves and thus fatty acids are the major sources of fuel. In addition, ketone bodies as well as lactate can also serve as fuel for heart muscle. However, the muscle prefer acetoacetate for glucose. Coming to the next organ, namely brain. This is exclusive organ which uses glucose as a sole fuel except for prolonged starvation period, consuming for example 120 gram per day, thus utilizing 60% of glucose by body at rest. If the level of glucose falls to 1 mm when plasma level falls below 2.2 mm, it is a danger signal. During starvation, liver supplies ketones to brain that partially replace glucose. The reason for non-availability of fatty acids as fuels for brain is that they are hindered by blood-brain barrier due to their binding with albumin. Coming to the organ that is adipose tissue. The tissue has the ability to store the triacylglycerol as a metabolic fuel. The organ is specialized for the esterification of fatty acids and their release, as you see here in the table. In humans, liver serves as a major site for the synthesis of fatty acids, and you can recall that the liver esterifies the fatty acids to glycerol phosphate, which is transported to adipose tissue as lipoprotein particles and esterified in the liver to glycerol phosphate to form trialysyl glycerol and are transported to the adipose tissue as lipoprotein particles. For example, as very low density lipoproteins. Adipocytes do not uptake the triacyl glycerol unless they are hydrolyzed by extracellular lipoprotein lipase, which are stimulated by the primary signal, namely insulin. The primary task of adipose tissue is to activate these fatty acids and transfer the active CoA to glycerol in the form of glycerol phosphate. It is known that the glycolytic intermediate dihydroxyacetone phosphate is reduced to form glycerol 3-phosphate. Hence, it could be perceived that the adipose tissue do require glucose for synthetic activity. If glycerol 3-phosphate is in excess, most of the fatty acids are re-esterified or released into plasma if glycerol 3-phosphate is limited due to dearth of glucose. This once again proves the fact that the level of glucose in adipocyte determine the fat of fatty acids to get released into blood, as shown here in this figure. Coming to another organ, namely kidney, though making up only 0.5% of body mass, kidney uses 10% of oxygen used in cellular respiration. Sodium glucose cotransporter driven by sodium potassium gradient, which in turn is conserved by the sodium potassium ATPase, helps to reabsorb almost all of the glucose. During starvation, gluconeogenesis by kidney contribute to 50% of the glucose in blood. Coming to linking the pools of storage, what happens here? The control points of metabolism are up and down switches and should not be misunderstood for the entire off-on conditions. It should be borne in mind, students, that there would be a flow of flux through a pathway even when the pathway is off. The major objective of integration, as we had seen earlier, is constant supply of glucose and energy. They are obtained directly from the diet generally, and if not obtained, the carbon flux changes to extract from the pools of storage. The Cori cycle that is shows in the figure and alanine cycles as shown here, the ketone body synthesis and utilization are also shown here. All these increased nitrogen metabolism and adaptations which require cooperation between various types of tissues, thus enhancing flow of glucose and energy during various physiological conditions. So let us see the first stage as starved, fed and well fed, that is post-absorptive state. Starved fed cycle has 3 stages: the post-absorptive state, the early fasting during night and the refed state, that is after breakfast. During all of these stages, the main goal is constant level of glucose, that is maintaining glucose homeostasis. During well-fed or post-absorptive state, amino acids along with glucose are absorbed from the intestine to be released into the circulation. The dietary lipids are transported by the lymphatic system packaged as chylomicrons. The primary signal insulin has a role in this state and stimulates storage of fuels as well as synthesis of protein in liver and muscle. It activates protein kinase cascade for glucogen synthesis for later use and promote glucose uptake in liver for conversion into glucose 6-phosphate. In adipose tissue, glycerol 3-phosphate is synthesized in this stage. So coming to the stage as early fasting state. Insulin and glucagon decrease and increase respectively in this stage. In liver, the main target of glucagon, it activates phosphorylase, inhibits glycogen synthase, lowers fructose 2,6 biphosphate and also promotes gluconeogenesis. It also inhibits fatty acid synthesis by attenuating the pyruvate levels and the activity of acetyl carboxylase. It releases fatty acids by adipose tissue and makes a shift of fuel usage from glucose to fatty acids. In brain, glucose gets oxidized completely, demanding alternate carbon sources. Following lipolysis, glycerol is released from adipose tissue to provide some carbon. Coming to the refed state. In refed state, fats and glucose meet a different fate. While fat metabolism proceeds same as that of normal fed state, glucose is not absorbed by the liver. leaving it in peripheral tissues. It continues its gluconeogenic action until the replenishment of glycogen stores, then only switches to the processing of excess glucose for the synthesis of fatty acids. Ketogenesis occurs in liver by the process in which 3 units of acetyl-CoA are condensed. Initially, 2 units form acetyl-CoA, which further condenses with yet another 3rd unit of acetyl-CoA, producing HMG-CoA. This is acted upon by HMG-CoA lyase, which produces acetyl-CoA and acetoacetate. Ketogenesis thus initiates with condensation and finally ends up in liberating 2 units free CoAs, which are locked as acetyl-CoA. Ketones are used by tissues other than liver by the conversion of acetoacetate to acetoacetyl-CoA, followed by conversion of acetoacetyl-CoA to 2-acetyl-CoA, which would subsequently burnt in the mitochondria of muscle. To conclude, this module presented here would provide a clear insight about the general and crucial features of metabolic integration. It provides a vivid roadmap of the major metabolic pathways with the key regulatory enzymes involved whose activation or deactivation based on the environment decides the course of metabolic reaction. This lesson highlights the influence of the enzymes whose presence or deficiency and also the shuttling mechanisms for the metabolites in various metabolically active organs in mediating interdependence and also subsequent energy maintenance. Further, the module presents the interconnectivity of stored energy reserves and also about the metabolic status during the different states of food intake, maintaining the energy demands.